Yeah, thank you very much. I'm happy to see you all back. So apparently I haven't scared you off too much yesterday. So we will basically just continue where we stopped yesterday. Let me briefly pull up the agenda again. So yesterday I talked about gene expression in general, some introduction to the central dogma. And then most of my lecture yesterday was about accuracy and speed in processing the genetic information. So copying mostly of sequences. What I want to do today is maybe a bit more general and also a bit more abstract. And I want to talk about a general approach to stochastic dynamics on networks, because that's, from a theoretical point of view, what these things are that we discussed yesterday. These were simple examples for networks where we have some stochastic dynamics going on. And then in the last part of today's lecture, I will talk a little bit about coarse graining. And just like yesterday, this will be a mixture of things that are tutorial, things that are well known for a long time, and I will basically just tell you material that you could also read up in the literature if you wanted, and some things that is our own work in the last years. So in particular, we have done some work on coarse graining, which I want to tell you about today. Okay, and those of you who just have heard the last presentation, you can maybe already guess that the part that will come tomorrow will have some similarities with what you heard in the last presentation, namely this more economic perspective. What are the costs of making protein in the cell and so on? So I think that will nicely follow up on Ajit's presentation, but this we will do tomorrow, not today. Okay, so our topic is stochastic dynamics on networks. And to start this, let me show you this graph again. No, this is not this graph. There is a little bit of an introduction here. So I want to briefly remind you that cells contain lots of molecular machines. And of course, those of transcription and translation, the ribosomes and the RNA polymerases, these are examples that I've talked about yesterday. Here you see an RNA polymerase. But there are also many other molecular machines, and the general theoretical framework that I'm presenting today can be used for all of them. And in fact, the examples that I will show you will, some of them not be from ribosomes and RNA polymerases, but rather from molecular motors of the cytoskeleton, because that's a system that has been very well studied, and it's also nice and simple to illustrate some of the points that I want to make about the general theory here. Anyway, so these molecular machines, they have many different functions in the cell, and this is something that you should all know by now after 1.5 weeks here in this meeting. Transport in the cell, information processing, and so on. And the one point that I want to emphasize here is that these machines have two aspects. There's, on the one hand, these machines all show some controlled large-scale motion. Okay, that's what gives us the right to call them machines. That's what machines do. There is some controlled motion there. Okay, and that's what we have. I mean, we have this controlled stepping of RNA polymerase on DNA, or the controlled stepping of kinesin on a microtubule, or in the rotary motors, you have a controlled rotation motion. So there is a controlled motion over relatively large distances. So in the kinesin motor, you have this one head going in front of the other. This is a 16 nanometer motion. So this is relatively big on the protein scale. And it's controlled. It's going from one defined position to a new defined position. So that's what makes us call these things molecular machines. There is some controlled motion. But of course, at the same time, these machines are molecular machines, and one of the consequences of this is that even though this motion is controlled, it's also subject to noise. Okay, so you see all these nice movies, how these motors work, how kinesin walks along a microtubule, how a ribosome processes messenger RNA and copies the sequence into tRNA, and everything follows very, very deterministically. And that's, of course, all nice, but you have to imagine that there is a huge amount of fluctuations on top of that that's never shown in these movies. Okay, so this just to keep in mind with all these machines, we have both aspects. We have controlled large-scale motion, but on top of that, we also have fluctuations, and these can be large, and often they are not shown. If you do molecular dynamics simulation, then you know why the movies of your simulation don't look like these illustration movies that people make of the mechanisms of these proteins, because the fluctuations are usually left out. Okay, this is the graph from yesterday that I wanted to show you again. So this is what we talked about yesterday. We have an enzyme, say an RNA polymerase. We have the new substrate come in, and well, in this simple model, this is reversible, and then we have some chemistry happening, and the substrate is incorporated into a new polymer. And one thing that we have used yesterday to reduce this, in that case, to this simple two-state model that I drew on the blackboard, is the fact that the enzyme at the beginning and at the end of the process is in the same state. Okay, the enzyme does its job, and then it returns to the same state. And that's also one of the aspects that is characteristic for machines, because the machine has to do something, and then you want to use it again, and it has to do the same thing again and again. So typically, all thermodynamic processes that are of some real-world interest, they work in a cyclic fashion, because at the end, the machine should be in the same state as in the beginning, and you can repeat the process. And this is what you see here also. Well, you don't maybe see it that well, because in the end, we go just from one state to another and kind of back, but really we go forward. But if we had two states here, going from the free state to a state 1, and then a state 2, and then back to the free state, then you would see that the enzyme goes through a cycle. You go from state 1 to 2 to 3 to 1, and that's a cyclic motion. And that will be important in what we're talking about today. Okay, here you see one of these examples with intermediate states, and I have picked the ribosome. And this is from a study from our structural biology colleagues, where you see that they resolve all kinds of intermediate structures. In this case, they differ in the configurations of the tRNAs. Here you have the tRNA in the E site, so in one of the 3 binding sites where the ribosome can have a tRNA that changes its configuration, and then they both tilt here. So you have this large number of configurations, and this is only a small part of the cycle. So this illustrates 2 things. First of all, the picture that I've shown you yesterday with this very simple 2-step reaction, that's of course really minimal. There are many intermediate steps, so this really consists of many sub-steps. But you also can see that if this runs around, then you get this cycle, and the machine at the end is in the same state as in the beginning. Here's another example. This is for the kinesin motor, where you have this stepping, and you also see that there is a cycle of states through which this motor typically goes to step forward. So every step it takes on a microtubule completes one cycle where it goes through a number of different configurations and different chemical states in terms of whether you have ATP or ADP bound to the motor. Okay, and this is our minimal model of a cycle. We need 3 states to see that it's a cycle. If we have only 2, then it could also be a back and forth. But if we have 3, then we can get a cyclic motion here. And that's our minimal model for today. But in general, we will look at some models that are more complex than this minimal one. Okay, here are a few more examples. This is something from our own work a few years back, So if you take one head of a molecular modus, you have minimally this 3-state model. If you couple 2 of them together, as in kinesin, where you have the 2 heads that can both go through a cycle, then you get something more complicated. And if you couple more of those, then you get a huge network that has many, many states and many different transitions between these states and many possible cycles it can go through. Okay, so I have just told you cycles are very important to understand these motors and any molecular machines. And this is our simplest cycle that we could have. But typically, in many molecular machines, we don't have just one cycle, but we have several cycles. So there may be something that is the dominant cycle, the typical working cycle of the machine, but there are additional ones. And these additional ones may have a specific function. And one of them we talked about yesterday, that's proofreading. Okay, so you have the normal cycle where a tRNA comes to the ribosome, the amino acid is incorporated into the new protein, and the ribosome returns to its initial state. But we could have an alternative cycle where in this middle state of that transition, of that path, when the tRNA is there, the ribosome does proofreading, decides this is the wrong tRNA with the wrong amino acid, and then returns to the beginning. So you have a second cycle that is different from the main cycle. In that case, this really has a function. But of course, there could also be another cycle that does not have a function, but rather that's something that happens because of fluctuations. There may be a mistake, and this gives another cycle. And that's what we have seen yesterday with the lower branch of our path, when the wrong tRNA, the wrong substrate is incorporated. That's also an alternative cycle. Well, it's basically just the second version of the main cycle, but if in the way we have drawn it, this was an alternative cycle. Okay, what's important in all these cycles, and this we'll see more later, is that they typically have some coupling to some energy source, typically ATP, because that allows us to get this cyclic motion in a certain direction, which is what these machines need. And the other thing that we will get to is that I should have shown these things while I was talking. Okay, you see some alternative cycles. Here you see possibly chemical energy coming in one of these cycles. And then what we will also consider briefly is that we have force coupled to the cycle, because some of the transitions may be dependent on force. And that's, of course, important when you have these large scale conformational changes where you have motion in space that could be hindered by a force. Okay. Let's start with the basics here. So the basics is that our stochastic dynamics in such a system is described by a master equation. I'm sure you have all seen this at some point. So you have probabilities for all the states, and you have transitions from one state to the other. And in this talk, I will use the convention that the first index is where the system goes to, no, where it comes from. No, sorry. It goes from state M into state N. So you read it backwards. This is somewhat confusing in the literature. One of them is easier to read, and the other is more consistent if you write this as a matrix element of a big matrix. And so today I'll use this convention that we read it backwards. Okay, so this is our master equation, and we can represent this, and in principle we have already done this. We can represent this dynamics as a network where each of these states is a vertex of a network. So these are here my 7 states, and then we have transitions between the states that are give us the edges in this network. And importantly, the edges here are directed edges, so between 2 states I have always 2 edges with different directions. In principle, I could have only one if the rate of one of the transitions is 0, but for what we'll do now, we assume that all these edges are there, and if one of the rates is really zero because a transition is irreversible, then we will assume that it's very small and send it to zero at the very end. Okay, so we can represent these stochastic dynamics by this network. And one thing that is very nice is that we can use this representation with networks, or mathematically speaking, with a graph to construct the steady state of that dynamics. Let me briefly show you the equation again. Of course, this equation is just a linear equation, or a system of linear equations. So in principle, this can be solved. So this graph-based method that I'll show you now, this is not because it's particularly convenient to use that to solve the system. You can just solve it. This is if you want, this is a large matrix equation. You can just solve that. But it's very nice to, a nice constructive way of getting the steady state in this stochastic dynamics. And this is very old. This goes back to work by several people in the 70s. And I think it has also been discovered several times over the course of the last, I don't know, 100 years maybe. In the end, it's also closely related to electrical networks that were considered by Kirchhoff in the 19th century. So if you want to, this is really not very new. Anyway, so what we're doing now is we want to construct the steady state solution of this system, and I will only sketch how this works in this graphical way. So if we have a network like this, the first step is that we have to construct the spanning trees. Do you know what a spanning tree is? Well, I think if you not, that is not representative. Sorry about that. Okay, so a spanning tree is a network that has as few edges as possible. First of all, the edges are not directed. As few as possible to connect all the nodes, and then whenever you leave out one of the edges that's still there, then you lose the connectivity. Okay, so you have the minimal number of edges with which you can connect all the nodes in this network. And of course, there are many ways of doing this. For example, this is one spanning tree for my small network here, where I have just left out this one edge. But of course, I could just as well have left out this one or this one. And then I would have other spanning trees. So what I need to do in this first step is construct all of them. And then in the second step, I construct something that has the strange name of an arborescence, which is basically a spanning tree that is directed towards a specific node. Okay, this spanning tree has no direction whatsoever. So what I do now is I chose a node I, let's say this node, no, in the example it's this one, node 4, and then I direct all edges towards that node. Okay, and for this arborescence, which has now two indices, nu and I, because I can do this for every spanning tree and then direct it towards every node of that spanning tree. Now, each of these directed edges corresponds to a rate. Yeah, so each of these corresponds to a rate, and what I do is I just multiply these rates. So all the rates that are in this directed spanning tree, these get multiplied, and then I take all these and just sum them up. So this omega, I now sum up over all the possible spanning trees. Then I get an omega that has only one index, I. And if I normalize this by summing also over the index I, then I get the probabilities of the states in the steady state of my stochastic dynamics. Okay, I showed you how to construct it, but I didn't show you why this works. And it turns out if you do that, it's really not very instructive to do that. You can take this, and if you put that into the master equation, then you have all these terms here which have all these different products. And when you insert it into the master equation, the probabilities pi, they all get multiplied with a rate. That's what we have on the right-hand side of the master equation. Sorry, here. Okay, these are my p's, and they get multiplied with an additional rate. And basically, what you have to then check is that everything that has an additional rate here gets cancelled out by another spanning tree where this rate is already in here, and the rate that was in addition in the term here is now the one in front there. So for every term, there is a corresponding term that cancels out, and then you know that this is a steady state. Anyway, so this is very nice because you can really construct the steady state of the system. If you have a large network, it's not necessarily practical because that's a lot of work. I mean, you have to determine all these spanning trees, and then, I mean, summing is maybe not so the problem, but all this enumerating of spanning trees, that's a lot of work if you have if you have a very large network. But conceptually, I think this is really very nice that you have this graph-based method of constructing the steady state here. And one can actually do the same thing for the fluxes. So if you take the flux between two edges, then that's just rate times probability minus the backward flux. And then you can do basically the same thing. I don't go into the details here, but what you get then is a sum now not over these arborescences, these directed graphs, but rather a sum over cycles in the system, because flux can only really flow when you go through a cycle, and then you have this sum over forward minus the backward products of rates in the different cycles of the system. And then there is a decoration, because if you have a cycle, this here is a cycle, there's also something like this, something attached to the cycle that itself is not a cycle, and this gives you the additional terms here in the end. So one can construct in this way a lot of properties of these stochastic steady states. Yeah, there's some literature in case you want to read more. The book by Hill is really very nice and easy to read. If you want to have a more detailed mathematical description, there may be this review by Schnarkenberg. It's good. And we have also written something in an appendix of a long paper on that. Anyway, so one additional property that I wanted to briefly mention is that there is, well, we have now constructed this as a steady state, but of course there is a dynamics that will describe how the system goes towards that steady state. That's what the master equation will describe. And one way of describing this is with this quantity here, which is the Kullback-Leibler divergence of the distribution at time t, so some pi probabilities of the states with the distribution pi0 in the steady state. So if you think of this as the distance between the current probability distribution and the steady state distribution, then this is something that should decrease over time and become zero when you reach the steady state. And that's exactly what it does. So this is what one can show, this property under the dynamics given by the master equation. It decreases, so it has a time derivative smaller or equal to zero. The time derivative will be zero once you have reached the steady state. And then also this k will obviously become zero because the logarithm here, this is a logarithm of 1 then. Okay, so we have a property that describes this approach to the steady state. And one interesting question is, what is this k actually? What kind of a property is this? Do you have any guesses what that is? No? Entropy production, interesting, yeah. Not quite. We'll come to entropy production later. Excuse me. Any other guess? Well, let's see. Okay, so this is this quantity K, and one way of looking at it is that we consider a system that goes into a thermodynamic equilibrium, because if a system is in thermodynamic equilibrium, then the steady state is simple. Okay, if this is say a canonical system, then in our steady state the PIs are just given by Boltzmann factors. And well, if I insert this in there, then I can pull this out of the logarithm here, and what I get is this expression. Okay, I have pi logarithm pi plus pi ei. Do you recognize this? A Shannon entropy, yeah, there is a Shannon entropy there, but there is not only a Shannon entropy. What is this term here? And the average energy, yes. So you have an average energy minus a Shannon entropy. Okay, so this is simply a free energy. It's a free energy. Well, it has somewhat strange units. There are some KBs not there, and it's a dimensionless free energy in some sense. Maybe I should have removed this KT to start with, so things are a bit simpler. But anyway, okay. So this is a free energy, and the free energy decreases, and when the system reaches its steady state and it's in equilibrium, this free energy is in a minimum. So that's nice. Okay, if we now go away from thermodynamic equilibrium, if our steady state is not an equilibrium state, then we could just say, well, this is still something like a free energy. And, well, I then just call it a generalized free energy. It's a quantity that takes the properties of the free energy outside of thermodynamic equilibrium. And in principle, we could say, well, the steady state here, instead of having this energy here, what we have is this steady state probabilities pi0. We could just say, let's take this equation. Now our pi0 is not in equilibrium, so it's not really a Boltzmann factor. But we could say, well, we use this equation to define an effective energy of each state. And then this is exactly that. So this K is a free energy, and if we use it in equilibrium, it's really the free energy. If we use it outside of equilibrium, if our steady state is not an equilibrium state, then we just have to interpret logarithm of pi, of the steady state probabilities as an effective energy. And then it still works. I think that's quite nice. Okay, so one more thing we'll need to discuss now molecular machines is detailed balance, and I hope that most of you have heard about detailed balance before. That's also an important property of equilibrium, that we have in equilibrium in every transition a balance that the flux in the one direction is equal to the flux in the other direction. Okay, that's this one here. And if I rearrange this, I get the ratio of the rates is equal to the ratio of the probabilities. And since in equilibrium the probabilities are these Boltzmann factors, then I get this constraint that the ratio of the rates is given by an exponential of the energy difference between the energies of the two states. Okay, and this is very important because it's a constraint on rates. So if you have one such network and you need to determine rates, you don't need to know all of them. Some of them you can get from this constraint because they are related to the reverse rate. And if you measure them all, then this might be something to check for consistency. Okay, if we use this in a cycle, then we can just multiply these ratios along a cycle. The energy differences will add up, and since eventually you end up in the same state where you started, the overall energy difference is 0. So we get that the product of forward and backward rates, the product of the ratios of forward and backward rates along a cycle, this has to be 1. So you can think of this as a global version of the detailed balance condition. So if this is valid for every cycle, then you have detailed balance in the whole system. Okay, and if we're not in equilibrium, so remember I assumed here we are in the steady state in thermodynamic equilibrium. If we're not in equilibrium, then these ratios will not be 1, and then along a cycle I can assign everything that's here on the right-hand side, and that's not 1, as a driving force for the cycle. Something that drives the cycle in one direction, and typically one calls this a chemical energy, chemical potential if this driving comes from chemical energy, which is the case that we usually have in molecular machines. Oh, what is this? That is not good. Okay, let's see what's going on here. Okay, let me check if I stop the presentation, if that's actually. Okay, this seems to have gone wrong when I moved from one computer to the other. Let's do the following. Sorry about this. We'll just go back here. I will do this on the blackboard. But I need my other computer as my notes. Sorry about this. So now we have to improvise a little bit. Okay, whatever happened to these things? Okay, so what I wanted to see here is our 3-state cycle. 1, 2, 3. Let me check my notation that this is consistent. Okay, and the idea is that going from 1 to 2, so these edges always have both directions. From 1 to 2, we have the ATP come in, so the source of the energy. And in the state, in the transition 2 to 3, we have ADP and the phosphate, so the two products of the chemistry come out. And then our third transition, this is where the mechanics happens. So this is the actual step. Okay, this is where we have the large-scale motion. That's my now minimal model, 3-state model for a cycle of a molecular machine. And now I want to use the detailed balance condition and some additional assumptions to put some constraints on this equation that I just had, where we have this product of rates along the cycle. Okay, and the first thing that I do is I just consider the step. I assume this isn't there. I just have the transitions between 1 and 3. Okay, So I have a forward transition and a backward transition. And if I have only these two, then they have to be related through detailed balance. And the energy difference between these two, that will depend on a force that's applied to the system. So whenever I have this large-scale motion, I do this against the force, then I basically walk up some energy, some gradient. Okay, so here I have a constraint that says that my rate omega 3,1 and my rate omega 1,3, these are related. And okay, here I've done it in the other way, but that doesn't matter. Let's write it the other way. 1,3 equals omega 3,1, and then we have a Boltzmann factor with the energy difference, and the energy difference corresponds to the force, if I have a force here, and the size of the step over kT. Okay, so in this direction I have the step, and force will increase the backward rate or decrease the forward rate. That's the first thing. The second thing that I want to do with this system is that I have the rates here and here that depend on molecules that come in. Okay, so my rate omega 1, 2, this is the rate with which I go from 1 to 2 and with which where the ATP comes in. This has to depend on the ATP concentration. And the idea is that, well, the assumption we make is that this is proportional to the ATP concentration. That's sort of the simplest thing we can assume. And I will call this now the rate without the concentration dependence kappa. So kappa 1, 2 times the ATP concentration. And to make my kappa, again, having the units of 1 over time, like this rate here, I divide my ATP concentration by some reference concentration, ATP 0. Okay, this is just my units of in which I measure ATP concentration. And of course, the backward rate in that case, this from 2 to 1, this will not depend on concentration because that's just one molecule of ATP sitting there in a binding pocket and that will go out. Here, the forward rate is the same. I also have now the products come out. So this has no concentration dependence. But if I go in the opposite direction, then the products have to come in. So there I get a concentration dependence as well. And I make exactly the same assumption here. So I have an omega 3,1. This I write as a kappa 3,1. And then product concentrations ADP, phosphate, and I divide this by some reference concentration. ADP 0, phosphate 0. This is not right here. Yes, this is all wrong. This is wrong and this is wrong. Okay, so this is going from 3 to 2. Okay, so this should also be 2, 3. Thank you. That's the danger of doing things on the blackboard. But they're also easier to correct than up there. Okay, so there's one more thing that I will do, and that will be convenient later, and that's that these reference concentrations, let me take a color here. So I have a reference concentration here, and here, what I use there are the concentrations that I have in an equilibrium of the reaction. Sorry? It's 2, 3. 2, 3, which means transition from 3 to 2. Okay, you always have to read the indices backwards. The first one is wrong. No, this one is right. Oh, these are, no, this is right. This is going from 1 to 2. This is this transition here, from here to here, where the ATP comes in. And then here, I have to take the backward transition from 3 to 2. So the 2 must be the first index in both cases because both transitions go towards here. Okay? I should have used the other notation because this reading backwards is very confusing. But it helps if you write this as a matrix because then these things really become the matrix elements. And otherwise you have to invert the indices when you write it as a matrix. So there is no good solution here. Okay, any more questions on this? No. So the one more thing that I want to do is I say these reference concentrations, which could be anything. I mean, they're just there to make this thing here dimensionless, and then this thing in front, the kappa, has the same units as the omega 1 over time. But what will be convenient is that I choose these as the concentrations in a chemical equilibrium. You can probably not read this. Okay, so this is what I choose. And now I need some space. Let's go over to this side. Well, maybe we can check whether this slide works, but I think it will not work. No. Well, let's leave this. Okay. So now I want to use this, and since we have this one cycle, I can apply my product that I had before. Okay, so I have this product over the forward rates, ij omega ji. And well, what do I have to do? Now I cannot see my cycle over there. Maybe you can tell me what is in the forward cycle. I have the transition omega. What are the 3 terms I have here? 1, 2, like this. Omega 2, 3, and 3, 1. Okay, and then here we have omega 1, well, 2, 1, omega 3, 2, and omega 1, 3. And now we have to insert these things from over there. And let me check. I think I need to sort the terms in some way so that this looks reasonably nice. Okay, the first thing we can make use of is this condition here. Okay, we have the local delta balance in the step where I have directly one rate, a relation between the 2 rates, which is this factor here. So which one was that? 1, 3 and 3, 1. These 2 terms, they will give me an exponential of the force. Okay, so let's write this here, e to the minus FL over KT. And then what do I have? I have 2 omegas left, I think, and 2 of the rates, the omega 2, 1 and omega 2, 3, they will get these, will become these terms over there. Okay, 2, 1 and 2, 3. These ones are still there. So what is left is the omega 3, 2 divided by omega 1, 2. And then for the other 2, omega 2, 1, 2, I get a kappa 1, 2, and here I get a kappa 3, 2. Okay, so that's what we have, and now my space is not big enough. But So what's now left are all these concentration dependencies. So this is also still there. I now have this times ATP over ATP 0. And I will write this. No, let's put this down here, okay? I have here ATP concentration, and I have ATP 0. Let me sort this in a way, and here I have ADP. And phosphate, and I have ADP 0 and phosphate 0. Okay, and this is not here anymore. Okay, now is where the thermodynamic equilibrium for my reference concentration is useful, because this ratio here I can relate to the chemical potential. Sorry? Everything fine? Okay. So this is related to the equilibrium constant and to the chemical potential. So I can write this as an equilibrium constant K times, well, the logarithm of that would be related to the chemical potential. So I can write this as E to some delta mu over KT. And I think now we're already done. Let me check if we did everything right. No, that's not right. This is not quite right. This is just the equilibrium constant. Sorry. Okay, this is just the equilibrium constant ratio of concentrations. Now, how does the chemical potential come in? The chemical potential comes in if we also take this term here. Because you need the, well, actually, if you are in equilibrium, then the chemical potential is in the minimum, and there is no driving force towards that equilibrium anymore. Okay, but if you have concentrations that are different, that's when the chemical potential comes in. So I can write this, now the logarithm of this one, that would be related to the chemical potential. So I can now write this term. If I am in equilibrium, now let's assume we don't have the force, so really we go into equilibrium, then this would be related to the chemical potential. So I can write this term here now as, well, e to the delta mu times 1 over k, I believe. Is that right? Yes. Yes, that should be, well, there's a KT missing. Okay, let's take a color for that because it gets too messy. So this ratio of the concentration, this is equal to 1 over the equilibrium constant times E to the delta mu over KT. So this thing will drop out eventually. And if we now have equilibrium, we don't have a chemical potential, we also don't have a force. Then I told you earlier that this product of rates forward and backward, this thing here, this has to be 1. Okay, but if we set the force to 0 and we set the chemical potential to 0, so this will be gone, but this is still here. So what we learned from this is that in that case, this also has to be 1. And these 4 things are just constants, so they have to be 1 exactly. So what are we left with? Basically for our whole expression here, we're left with just e to the delta mu minus force times L divided by kT. Okay, that's what we have. So in our simple model, in our three-state cycle, basically we have this expression that the product, the ratio of forward rates divided by backward rates, that is given by an expression of this type that I've written in the equation before. But we have two driving forces here. We have a chemical driving force that goes forward, and we have a mechanical driving force that goes backwards. So this is basically the longer version of this equation that in the last slide that worked. I just told you everything on the right-hand side that is not one, we call it a chemical potential. In this case, we don't call it all the chemical potential, but we have a combination of chemical energy and mechanical energy that drives the system. Okay. Sorry for this somewhat messy derivation, but probably this has slowed me down, and we will go over time. Okay, so let me start the presentation again, because I hope from here on things will work. Okay, this is for a single cycle, and if you have more than one cycle, things become more complicated. And one thing that I don't want to go in detail, but there has been a discussion maybe, well, now 15 years ago or so, long ago, about the kinesin motor, whether there is one cycle or more than one cycles. And there are some indications that it can't be one cycle, because one cycle, a single cycle, is very constrained by thermodynamics. We have seen here that we have all these different constraints on the rates. And one thing you can see here is that if there is no flux in a single cycle, then basically you have to be in equilibrium. That's a very strong constraint. So if you have only one cycle and you have no flux in this system, then you are automatically in an equilibrium state. And if you are in an equilibrium state, that means you have, well, this thing here has to be zero, and that means whenever the force is such that the system doesn't move, the chemical potential has to exactly balance that force. So these two energies here have to balance, and that's a strong constraint, which gives, since the chemical potential depends on concentrations, that gives a relation between the force that you need to stop a motor and the concentrations of ATP, for example. And if you look at it here, for example, you see the kinesin. Now here you see kinesin velocity as a function of force for two different concentrations of ATP, and there's basically no dependence on that concentration, which, if you look at these relations here, can't be. There must be a relation if there is a single cycle. So that's one reasoning, one line of argument why there can't be just a single cycle. Because in a single cycle, everything that goes backward has to do the chemistry backwards and the mechanics backwards. So every backward step has to make ATP. And that's actually not true. That's another thing that was shown in this study by Carter and Gross in 2005, that kinesin, when it walks backwards, it still uses ATP. When your car drives backwards, it also does not produce fuel. Okay, so it's not terribly surprising that this doesn't work in this way, but if you had a single cycle, so your car is also not a single cycle machine. The single cycle is a very strongly constrained system. Anyway, and that's why, Liepel and Lipovsky at that time proposed this model, which has three cycles. So you have a forward stepping cycle in which ATP comes in somewhere. So here ATP comes in the step from 1 to 2. It makes a step, and the ATP is hydrolyzed, and so on. But in the backward step, you have also ATP come in here from 4 to 5, ATP comes in, it makes a backward step, and it consumes the ATP. So in this system, you have a forward cycle and a backward cycle, and both of them consume ATP. I'm showing you this because I want to use it later on as an example for a model that is slightly less simple than this single cycle model that I've shown you here. Okay, and there's the third cycle here, and that's the cycle where you never make a step. You only consume ATP. You consume ATP without making the forward step, and then you're here, and instead of making the forward step, you go through the backward stepping cycle, but you also don't step and just do the chemistry. So it's basically a machine that just burns fuel without doing anything. Okay, so we have 3 cycles here, the forward cycle, the backward cycle, and the surrounding cycle that is completely useless. Okay, that's the first part of what I wanted to tell you today. In the second part, I want to tell you about something we have worked on in the last 3 years or so, on and off, and that has to do with coarse graining. And almost everything I'm showing you today is the work of a master's student, very good master's student, who then unfortunately moved to Oxford after that, David Seifert. And what we were thinking about there is how can we simplify such a network? If we have a stochastic network of the types that I've discussed so far, can we make it smaller by leaving out details that are maybe not so important? Okay, and the way how we said we can simplify a network is by merging nodes. So for example, these 2 nodes, there are 2 ways of getting from I to J. Here in between, we could say, well, we merge these 2 together. And of course, when we do this, there are also other ways how one can simplify networks. There are other procedures for coarse graining networks, but that's what we have chosen. We have chosen to merge nodes. And then what we have to determine is what are the rates for this simplified network where 2 nodes are merged. Okay, and there's 2 ways of determining the rates. One is very intuitive and one is more formal. I will show you the intuitive way. And the intuitive way has basically three conditions. The first condition is if we merge 2 nodes, then any rate that is far away from these nodes, that doesn't touch those, any edge that should keep its rate, should not be affected. Okay, so the tilde rates are now the coarse-grained rates. These stay the same if neither of the 2 nodes here is one of these 2. And then the second condition that we have is that if we merge 2 states, the probability of these 2 states should just be added up. Okay, that gives us a second condition. If 2 states are merged, then the combined state should have just the sum of the 2 probabilities. And the second condition is that the flux from the merged state into this one, for example, going from the combination of these 2 to I, this should be the sum of the 2 fluxes that we had before. So that the total flux that goes back towards I stays the same in this coarse graining step. And then these conditions give us simple expressions for the rates. So rates going out of the rates going out are simply the sum of the rates going out of the 2 individual states. This is this condition. And the rates going in, they are a combination here, which unfortunately depends on the probabilities of these states m and n. So this is bad in the sense that when we want to use this to simplify our system to be able to calculate things easier, then it doesn't help us. Because for simplifying the system, we already need to know what the solution of the complicated system is. So that is maybe not super helpful, but of course we could say, well, what we need is actually only the steady state of the complicated system. We can then simplify and do dynamics of the system afterwards. I don't find this argument very convincing, but I think what is still useful for, even though practically there is this problem here, is that this coarse graining tells you something about the conceptual understanding of such a system. So one thing that we actually are working on and is to replace this with some estimator that doesn't require us to know these rates here. Debashish, you have a question. Yeah, that's a very good comment. So should this transition be much faster than these? Yes and no. So we can do this procedure without that condition. Okay, the procedure can always be done, but of course what you can say is if this transition is much faster, then the error we are making in doing this procedure will be smaller. But the procedure can be carried out independent of any consideration of transitions being fast or slow. It's always doable. The question is how much information do you lose when you do it? And if this is very fast, then you lose less. Okay, one nice feature of this procedure is that you can do this iteratively. So you can merge two nodes and then you can merge more nodes. And you can do this as many times as you like, and the result will be independent of the order in which you do this. This is not true for all procedures that you can imagine. There are some procedures where the order in which you simplify things matters. But in this one, it doesn't. There is a second way of deriving these rates, and I will not go into this, but in that case, you can take the probabilities of trajectories in the two models, in the original model and in the model where you have merged some nodes, and then again consider the Kullback-Leibler divergence between these probabilities. And what you can do is minimize this, find rates that minimize the Kullback-Leibler divergence. So basically find rates in which you modify probabilities of paths on your network the least. So you try to make a very small change in probabilities of paths. That gives you a condition on the rates, and it gives you exactly the same result. You can generalize this even more, and that's also something we're working on right now. If you don't use the Kullback-Leibler divergence, but some other measure of this type, and then you get different results. You also don't get this nice feature that the merging of nodes is independent of the order in which you merge nodes. So in some sense, this thing that agrees with my intuition, this is very nice, and the other features are maybe theoretically more appealing, but they also have some features that are not so nice. Okay, now let's apply this to a system, and I have now a network of 6 states, and if you look at this, this looks very much like this 3-cycle kinesin network that you have seen before. And what we do is we merge these 2 states and we merge these 2. So we don't change the topology of the network here, we just change the number of intermediates. And we compare 2 quantities. One quantity is sort of the velocity of the motor, so the overall flux through this rate 5-2. This would be the forward step in the kinesin model. And what you see is this is very nicely conserved. It's the same in the 6-state model, in the 5-state model where we have only 2 of them, well, these 2 are merged, and in the 4-state model where we have merged states on both sides. Okay, this is the same, and it should be, because that's how we constructed our procedure. We wanted the fluxes to remain the same. There are some other quantities that also remain the same, and there are some that remain almost the same, but there is one quantity that really does not stay the same, and that's the entropy production in this system. So this is plotted here. Entropy production is a sum over all the edges, and then for each edge we have the probability of the flux and the well, basically the affinity, the chemical potential here. Okay, this is logarithm of what is on the right-hand side of this type of expression if we use this for a single transition. Okay, if this is 1 on the right-hand side, then we have detailed balance. This is basically the deviation from detailed balance, which corresponds to a chemical potential for the chemical potential difference for the individual step. Okay, so this entropy production is this sum over fluxes times corresponding chemical potentials. And what you see here is this is very different. This is the original model, and in the first step where we merge these two, actually this doesn't change much. It looks like it does not change at all, but it's just the difference is numerically extremely small. It's not exactly the same. And the reason for that is that in this system, the rates here are such that this is a transition that is not so important. The system, if you think of this as kinesin, it would be a kinesin at low force that mostly makes forward steps. And if we change something in the backward step, it doesn't affect the overall behavior too much because this flux here is very small. But once we go from the 5-state model to the 4-state model, we change something in the forward step, and you see that in the entropy production here, we have a decrease of something like 10% or so. So we lose something by merging these states. Yes?
I mean, if I just look at it like a school student and use Kirchhoff's circuit, so essentially there are some conductors or some wires where there was a Joule heating originally, and now since you have removed them, that Joule heating has disappeared, and so that's why the entropy production is lower.
Yes, you could think of it in this way. I think so. It's not the way I think about it, but I think you could do that. Yeah. But that's I guess I don't have that much intuition for electrical circuits. But yes, yeah, I think it's a good way of thinking about it. Okay, so what you see here is with coarse graining, you do lose something. It's not exact. You make a mistake. Some quantities stay the same, but there are some quantities in which you make a mistake. And one of the ideas we had when we did this is we said, well, in principle, we can do different things. I mean, merging these two, that didn't do much to the system. So merging these two states, that was something that made a very small mistake, but merging these two, that made a bigger mistake here. So what we thought is, well, maybe there is an optimal way how we can simplify the system. And what we then said is we simplify the system in such a way that we, on the one hand, try to take away as many states as we can, while on the other hand, at the same time, try not to take away too much entropy production. Because taking away entropy production, that's a signature of loss of important things. That's the idea. And so what we did here, this is again the same system. We said in every step of coarse graining, we merge two nodes, and we always merge these two nodes that give us the smallest decrease in entropy production. Okay, or in Debashish's language, what we take out is the one element that produces the least heat. So most of the system stays the same. And then if you want to, you can say we balance this and we say we drive this as far that sort of the loss in entropy production minus the decrease in number of nodes, that this is minimal. That's what we do here. So we remove the first, we merge two nodes, we merge another two nodes, and so on. And in every step, we lose some entropy production. This gets bigger and bigger. That's the blue curve here. In every step, we lose one node, so the orange curve here, that just goes up linearly. And if we take the difference of the two, there will be some minimum here, and that's in some sense, according to this criterion, that would be the optimal degree of coarse graining that we should do, where we can take away as much as possible, but not take away really crucial parts of the entropy production. Of course, that's a heuristic argument. You could do this differently. Again, what you see here is that we did this 3 times with different parameters, which correspond to different forces in this kinesin model. And if you just look at the graphs, you see in every step we took away one node in such a way that we had the smallest effect on the entropy production. And what you see here is that for small force or zero force in that case, basically first we merge these 2 nodes. That's what we have seen on the last slide already. And then we take away this complete backward cycle because the motor anyway goes forward most of the time. So we are stuck with, we only have the forward cycle, and then we can still merge nodes here. And in the last step, which would be really way too much in that direction, we end up with a 2-state system, which has to be an equilibrium system. So there you have lost everything that makes it a motor. But if we do the same thing at large force, let's take the very large force first, then you see that we first lose the forward cycle and keep the backward cycle, and then we merge things in the backward cycle, and well, at the end we go to equilibrium. And at the intermediate force, which is close to the stall force, there, well, things happen in both cycles, and basically we keep both cycles relatively long. So the optimum here is a description that keeps both cycles but simplifies both cycles into 3-state cycles. Okay, so here the optimum is this 1 3-state cycle. Here it would be the 3-state backward cycle, and here it would be keeping both forward and backward cycles but having them simplified. No, I don't use this computer, I use this one. Okay, if we now plot things as a function of force, then you see that the models now give very different results as a function of force, because we have just seen if we coarse grain at low force, we should actually use a different model than if we coarse grain at large force. And if I just plot all the models together, you see that, well, for small force, the difference in velocity is not so big. But if we go in this region near the stall force, you see that we have very different curves. The blue one here, this is our full model. And then if we just have the forward cycle, we go up here. And you see that the stall force is completely different. Because the stall force now, since we have only this one cycle, that's determined by the chemical potential, by the ATP concentration, and it's completely off from the real result here. And whether we have 3 states or 4 states in our one cycle, that doesn't make much of a difference. No, this one, the forward cycle. Whether we do this with 4 states or 3, that doesn't make a difference in this curve, but the curve is very different from this curve here. If we use this cycle where we have both, this model where we still have both cycles, but both cycles are simplified, then the curve looks pretty much the same as the original. And you can do the same thing for the entropy production. Down here, where you see here blue again is the original model, and then all the other models, they have lower entropy production, and well, depending on whether you are at high force or at low force. At high force, the red curve, which is this one, that's still very good. That has almost all the entropy production, while the others that have only one cycle, they have very much reduced. And at low force, most of the models are reasonable, but the blue one, the green one, which has only the backward cycle, that of course fails completely. Because now you try to describe a forward motion by a backward cycle that runs in the opposite direction. Okay, let's see. I think we're pretty much in time. At this point, I should come back to proofreading. I should tell you how we coarse grain the proofreading that we discussed yesterday. But unfortunately, I can't do that because we haven't done that yet. So this is something that we still have to do. Okay, I will tell you about this when I come next time. Sorry about this. But this also means that we can actually finish on time, even though we had this slight delay of me going to the blackboard. Okay, I will finish here. So what have we seen today? The key points are the following. I have told you in the first part that we have this network description of stochastic dynamics, discrete state stochastic dynamics, which gives a very nice way of visualizing this dynamics and also of constructing properties of this dynamics. I have tried to show you the connection to thermodynamic properties via the cycles where you have these detailed balance conditions or deviation from detailed balance that give you some of these thermodynamic potentials here. And then at the end, I've told you about we can coarse grain, about how we coarse grain these systems by merging states. And I've shown you that in this case, we lose entropy production, but by doing this in a way that the loss of entropy production is minimal, we can find a way how to coarse grain these systems in an optimal fashion. Again, how we do it at the moment is not a practical way of simplifying models that we can't solve. It's a way of simplifying models to see conceptually what's going on there. We're still working on how to make this a practical procedure for large systems. And then in the specific applications, I told you that the single cycle models are strongly constrained by thermodynamics. So a lot of the things that are observed in biological molecular machines actually require more than a single cycle because you see that these constraints are not fulfilled. And then you need multi-cycle models. And sometimes, as you have seen in the coarse graining example, you can go back to single cycles in some limits, low force or high force in the kinesin. Or if you think about proofreading, you have a proofreading cycle and a correct incorporation cycle. If you never incorporate incorrect ones because the concentration of incorrect monomers is very small, then you can probably coarse grain away the proofreading cycle because it's not used. Okay, this is where I stop. Tomorrow in my last talk, I will tell you a bit more about the molecular machine. So this will again be less theoretical and more biological, and then tell you about the economic principles, how cells use these machines, and then also talk again more about transcription and translation. Okay, so that's it for today. Thank you very much for listening, and I think we still have time for some questions if you still have questions. Thanks.